integrated program of study that crosses the science, art-making, humanities divides and culminates in a senior project combining at least two of the fields. Find out more about that program at sas.upenn.edu/visual-studies. Hugo Spiers also crosses the science and art-making divide. After earning his PhD at the University College London Institute of Cognitive Neuroscience, he held postdoctoral fellowships at the MRC Cognition and Brain Sciences Unit in Cambridge and the Wellcome Trust Center for Neuroimaging at UCL. He was then awarded the Wellcome Trust Advanced Training Fellowship to learn single unit reporting. In his lab now at UCL, he is senior lecturer in the Department of Experimental Psychology. He uses cognitive and behavioral neuroscience techniques to study the neural basis of spatial cognition, including imagination and memory. He has received grants from the Wellcome Trust to collaborate with artists in exploring these topics through art installations, which can be seen on his website. Some random facts about Hugo. If not a neuroscience scientist, he might have been a filmmaker, or an archaeologist, or a philosopher, or a physicist. Also, he is dazzled by the fact that during sleep, the hippocampus can replay its internally mapped journeys through space at 20 times faster than reality. Today's talk, entitled Brains on Show, Neuroscience and the Visual Arts, will be followed by a question period and reception. Outside. Please join me in welcoming Dr. Hugo Spiers.
Thank you very much. Just testing the mic. Thank you very much, Gary, for inviting me to give this talk. And I always start, like every talk you would imagine, academic talks, by that sentence, thank you for inviting me. But today I'm really extra thankful, triply, quadruply thankful to the team here because I don't normally get the chance to put the brains on show. Normally I talk about my brain imaging research or other parts of my research. And I'm going to do a little bit of that today. What I'm mainly going to talk about is a sort of secret hidden part of my life to most colleagues I work with at UCL in London, which is the collaborations I've done with the visual artists and filmmakers over the last 5-7 years or so. So let me give an overview of what I'm going to tell you in the next 50 minutes or so. First thing I'm going to do is be very egotistical. I'm going to focus pretty much on two particular installations that I've done, and collaborations I've formed that were funded. I'm going to present those, and after I presented those to you, I'm going to go through the creative design process. And in fact, to those neuroscientists in the audience, I would note that this is the reverse order of what we're used to doing. In most talks I talk about why we're doing these things, how we developed the experiments, what the experiments showed. I'm going to do the opposite today. You start with the outcome and work backwards. After that, I'm going to try to be less egotistical and talk about how this work I've done relates to other pieces of work that are out there in the field and collaborations. And finally, I'm going to move into a bit of new research that we've just finished conducting that relates to these topics. And that is the question of, do painters, sculptors, and architects think differently about space? And to describe that new work and then wrap up. So let's start with this beginning bit, the installations. So I'm going to show you two installations. And they were both initially shown in this location here, in Gimple Feast Gallery in London's Mayfair. And they were both funded by this charity, the Wellcome Trust. So part of the story, the narrative of why I'm here, fortunately here today, standing talking to you, is to a confluence of this charity, who described themselves as the global charitable foundation dedicated to achieving extraordinary improvements in health, supporting the brightest minds. Most of what they do, in fact, occurs in the City of London trading stocks on the stock market, and they slice off a little bit to fund a lot of fantastic neuroscience research and beyond that in immunology and health. But they also decided, very cleverly, I think, to slice off some of that to engage scientists with artists. And they realized that while scientists can go out there and give talks, they're not always the most engaging or colorful or wonderful, but in fact, artists do a much better job of engaging the public. And this occurred in the year 1999. They started a program which they've now increasingly funded more. The other key fact is this gallery, and this gallery forms a part of these installations. So Gimpel Feast Gallery, I ended up working with Gimpel Feast Gallery because I fell in love with a girl. I went to a conference a long time ago as a PhD student on how to get out of your PhD into the real world and do something useful with your life. And I failed. I'm still here in the academic system, but my partner, my wife, she was from this art dealing family, Gimpel Feast, and she did escape. She works in the media. But when I met her and I visited this gallery, I got introduced to a world of art, which I wasn't exposed to before. So Gimpel Feast Gallery, to those who, many of you may not know it, is situated right in the middle of Mayfair, the most expensive place on the Monopoly board, if you ever played Monopoly. It was founded in 1946 by Charles and Peter Gampel, who were the sons of the famous art dealer René Gampel, whose diary, A Diary of an Art Dealer, is a really important text if you're working in art history. But they've had many, many famous artists in this gallery in the past, and they've worked with Yves Klein, Marcel Duchamp, and many other artists. And it was a huge honor for me then to work with this gallery for the projects I'm about to show you, and collaborate with the team in there. So the first project that's set in this gallery that I'm going to take you through is called Neurotopographics. And neurotopographics, the name comes from the idea of brain, space, and diagrams and drawings. So that's effectively what this was. We named it this way because we realized cynically that the grant funding agency really liked funding things with mysterious names like this. But it stuck. So what was neurotopographics? So what I want to do is not tell you much more about it at the moment. I want to take you as a visitor to the gallery, take you back. I'm actually going to dig out from my book, the little piece of information you would have gathered if you'd gone to the gallery on the day this was being shown in Gimpel Feast. You'd arrived, you'd walked through that gallery space. You'd have to walk through here, down some little steps on the side, and you'd enter a little underground bunker where there's a very dark room. And inside the room, it looks like this. So on the floor, there's a huge floor projection, which you can stand on. So you can stand and walk over this floor space. And on one wall, there's a projected image of a white wall with some black lines upon it. And you'll also notice there's a speaker up here, and hopefully the sound I'm going to play in a moment. And you might have picked up this little information sheet on your way in and read, Gimpel Feast is pleased to present the groundbreaking exhibition exploring the boundaries of art, architecture, and neuroscience by exploring how mysterious patterns of brain cells allow us to perceive and remember space. The film installation Neurotopographics is the result of a Wellcome Trust-funded partnership between neuroscientist Dr. Hugo Spiers, artist Anthony Malinowski, architect Bettina Wiesmann. The exhibition will be accompanied by blah, blah, blah. You probably wouldn't have read much more beyond that, or at least I don't when I go to most galleries, unless I get really excited. What you would have then done is walk into the exhibition, and here's another view. So once you're standing there for a while, another image appears up on the wall. So we have two images, one on this wall and perpendicular to it, another wall. In this image, you can see a man standing in the space. And obviously, I can't recreate this huge floor installation in this room to really bring you into that. What I'm going to do is show you the website we generated that can show you footage from this floor projection and from this particular view. So I'm going to do that now, and hopefully it will behave. Let's go into. So here we are. So what you're going to see on the left here, in this black space, is footage of a man walking around in the room you would have just walked through in the gallery to get to where you are. On the right-hand side here, you're going to see a projection that would have run on the ground underneath your feet as you're standing there. And you'll hear the noise from the loudspeaker positioned there. Let me just... There we go. And it's about 2 minutes long. Okay, so that is the film, and it would have looped in the room. And what you have to imagine is standing on that white space with the film moving underneath you at this point. And this film you saw on the left-hand side with the man moving. Shown here is a changing view. Unfortunately, I couldn't put it, we didn't load it into the web page, but it follows his first-person view around the room. I'll come back to the importance of that later in the talk. So we showed this. This is the sort of setup in Gimple Feast. You can see the door where you would have come in, on perhaps on the side here. It's a small, intimate room. We showed it in that gallery. And I should add, just as a context, this is a commercial art gallery. So the week after we showed this, somebody had a show with black bin bags hanging on the walls, small bits of carpet, and some spray-painted twigs. I think each one of them was sold at about £1,000 because it was some well-known artist in a particular tradition, and these pieces of bin bags had been carefully collected. And it was very intriguing work, but it was the kind of space to take a neuroscientist into, and very surprising what would be shown there. So that was neurotopographics, and we also showed it. So here's me in a experimental gown that is necessary to wear under UK regulations in this room, and this is a room we also showed neurotopographics in. This is Anthony, the artist involved, and Bettina, slightly blurred, who is the architect involved. They're wearing special shoe covers because you're not allowed to step in the surface of this room without shoe covers on. And that's because this is an experimental room in UCL for researching spatial cells in the brain that represent space. And you can see now here the different films and the floor projection. This is a very different contextual setting, and I'll come back to that in this talk, like the importance of that. But this was at the opening of this new institute, and we had Joe Ledoux and other neuroscientists come over to open it and look at this piece of artwork. So that's all I'm going to say at the moment on neurotopographics. I'm going to move to my next project, which I did, and I'll come back to neurotopographics. After having completed neurotopographics, I was still interested to explore these cells, and so I applied for more grant funding from the Wellcome Trust and was very fortunate to receive it to do this project. So pattern completion was again initially placed in Gimple Feast in this small space under the gallery. So you'd have walked through the main gallery, gone downstairs, and entered that same dark room. No wall projection, no projection on the walls, this time projection into glass spheres. In this projection, and here's a zoomed in image, these are kind of this size in your hands. You could hold onto them like footballs. These glass spheres hanging suspended in the dark space with photographs, time-lapse photographs projected into the spheres. Here's another view of these spheres. You can see the projector beam of light thrown onto the spheres here. And as you walk around the room, you can see into the spheres, and you're also given headphones, a set of headphones to wear. And as you walk up to these spheres, you can hear the sounds of forests that are projected into the spheres. You hear the noise of peacocks or pheasants or different animals moving around. You might hear the noise of a river running by in the background. And you see the slow swaying of the leaves and branches. Here's another image of the forest scenery projected into them. And here's, just to give you a sense of scale, here's Tom, the sound designer who put together the sounds, looking at the spheres here. And here's another view. So here's the room. There were some headphones here, and there's a small little spheres hanging there in the space. And what I'm going to do now is just show you a short film of walking around this installation. So let's see if that plays. There's no sound accompanying this one. I apologize for the lack of sound. Yeah. Here we go. So this is a dark space. So we showed that in the gallery at Gimple Feast, where, again, commercial art gallery, but we also took it out of the gallery and presented it to the public at University College London. This is the main quad. This is the Slade College of Art in London at UCL. And this is the observatory, which is locked away to students who can't normally get in there, but somehow we managed to get the key and put this installation inside this observatory space and have visitors come and listen to the sounds and view those spheres in that small observatory, sorry. Okay, so what I've done at the moment is show you introduce these installations. I can't transport you to that space. If I could, that's what I would have done, but I've showed you them as best I can on those screens. What I really want to do next is to dive into the design process and think about how we generated those and why we generated those. Okay, so neurotopographics, coming to this project here, what we were trying to do with that project. Well, the project Neurotopographics and really the start of all the collaborations I've done with scientists had taken place, all occurred by this painting standing here, where I ran into this man, Antony Malinovsky. And I was at an art opening where he'd just finished collaborating with Michael Nyman to produce a piece of music related to his wall paintings. And he's an unusual artist, and he won't fit the bill for the end of this talk in the sense of he's not a typical painter or an architect or a sculptor. He does large-scale paintings like this. This is Antony painting on walls. And this is another one of his paintings. And the first thing that struck me as strange, back in 2004, had a whirl in my life in neuroscience. I rarely met artists. It's fantastic here. You have a program to allow that to happen. But when I met Antony, one of the strange things he said to me in conversation was that he was afraid of objects. And I thought, this is a very strange thing. I've never heard a scientist tell me they're afraid of objects. I don't think he literally meant he left his house and was terrified walking down the street with trees. What he was getting at is in a working practice, three-dimensional objects were not a pleasant thing for him. And most of his work plays on surfaces and the boundaries of space and ambiguities of what is an object and what is a background. And these, of course, is a fundamental topic that many, many artists have played with and used and explored. But it seemed to me that these kind of works are fantastic at that time. Talking to him, I thought, what if we can take some of the crazy ideas in neuroscience and throw them together with an artist that does this kind of work? We could make the damn best illustration of the science possible. And that was, of course, a mistake, to think that we could make a really great illustration of the science through working with an artist like Antony, as I discovered when I started the thing. But we initially decided to collaborate together and work on this science. So that installation, Neurotopographics, you saw, was motivated by work that a couple of months ago, or a month and a half ago or so, won the Nobel Prize for Physiology in 2014 this year. And the Nobel Prize was awarded to John O'Keefe, who was one of my mentors as a PhD student. It was also jointly awarded in half to him and half to May-Britt Moser and Edvard Moser, who are two Norwegian scientists. So John is at University College London, and they're in Trondheim. So what did they discover? What was it that I was so excited about that I thought, wow, we've got to turn this into some piece of artwork, because this is way more exciting to me than those sticks lying on the floor I had seen. I thought, I can do something better than this. I can do something with the science here. What John discovered in 1971, when he was recording from the cells in the hippocampus of the rat, was that, shown on this diagram, figure 1 here, as a rat shown in this little black dot, if it scurried all around a square box looking for food, he could record in black where the rat went. But if he recorded from an individual cell in the hippocampus, and a particular type of cell, pyramidal cells, in the hippocampus, it would burst activity. It would release action potentials that he could record through electrodes whenever the rat was in a particular place. So shown here is silence. The rat is running all through that space. But when the rat ran through here, John would have his loudspeaker on in the room, and I've done this, and I'll show you some of the data I collected. You have a loudspeaker on in the room with the rat running around, and you listen for these kind of bursty, punchy noise of the action potentials converted to sound. So, of course, they don't make any noise in the brain. But to be a scientist like John and Edvard and May-Britt, you have to tune your ears into the neurons. You can't do the work by looking at it. You have to tune your ear to the noise of the particular cells. And after a lot of experience working on these sorts of things, you get a real reward sense when you've hit the right neurons. But what John found, as shown in this diagram, were cells called place cells. He coined them as place cells. So these cells, John argued with Lynn Nadell in 1978, formed the basis of a map of space. So here we are in Penn campus, and the idea that these cells provide us, here's one cell, but each part of this box, a different cell will like that different part of the box. And as you walk around here, as you walk around this room, I've never been in this room before, but as I walk around this room, my hippocampus' place cells will become active. Right now, I have a set of maybe 10,000 cells that like this spot. And as I stand over here, they've turned off. A new set of cells, a wholly different set of cells are active. And as I move over here, a new set of cells. Everywhere I go, a different ensemble of cells like this will become active. And if I try to remember, maybe in a week's time, this lecture, hopefully nothing bad happens, but if something bad happened, I might remember back and reactivate those cells later. So these provide this map of space, much like a sort of external map, but these are the internal map of space. What May Britt and Edvard discovered that was so shocking and jaw-droppingly described at the time when it was released at the Society of Neuroscience meeting was that when they recorded a rat running around in a box, from a single cell, they found cells that fired much like place cells in isolated parts of the box. It's silent for a lot of the time, but in certain bits of the box, that cell will become active. And what they noticed, or somebody pointed out to them, is that these weren't random. They formed this beautiful grid pattern in that space. And these cells were coined as grid cells. And they provide something, a bit like a latitude and longitude signal. And just to show you, this is some data from the place cells here. I'm not going to show you any grid cells. But this is typically how these cells are plotted out, through black lines and red dots for where cells are active. And then you can take a rate map to show the peak hotspots of where the cells are active. And this is some work I did looking at how place cells represent rooms in a corridor. And we find that in this work that these place cells, they represent space, but they really like doorways. But the grid cells don't care about doorways. They provide this latitude-longitude signal. But crucially, they don't do it in the way our GPS system works. They do it in a hexagonal tessellating pattern. And I hope this, if you're using your place cells to remember the past, looks somewhat familiar to you. So the idea in neurotopographics was to take these ideas from these static images like this in the research papers and explore what if we could see those cells visualized on an architectural plan of space? What would it look like? And so I took from Anthony's work, he'd been etching into spaces in his artwork. Here's some more of Anthony's work. We thought, what if we could use this kind of manipulation of space in our installation? What if we could explore the patterning of space in our installation? And we worked with Bettina Wiesmann. At the time I met Anthony, he said, oh, this is great, this conversation, but would you mind giving a talk at the Association of Architecture in London? Bettina Wiesmann was holding a workshop there about microspaces. So we gave a workshop on that, and then this collaboration kicked off from there. And Bettina spent a lot of time sketching up, working out how is it that we could take ideas in architecture, take the ideas from Anthony's manipulation of space, and combine that with the theoretical work on place cells and grid cells to produce a piece of work. And one of the key innovations, I think, that I, working with the artist and the architect in this, was to think we need to think about delusions. We need to think about hallucinations. If we just take the data, and this is what every time I see anyone illustrate place cells or grid cells, they'll show it in the way we do in the lab. They'll take it. But to actually understand what these cells do, we need to look beyond that to these kind of strange things. So Lilliputian delusions are things that occur, I don't know if anybody's had Lilliputian delusions, but they're documented historically in many, many cases of writers. This is where people start seeing little creatures in the corner of their eye climbing the skirting and the walls. It recurs in lots of situations. These Lilliputian delusions, little characters running around in the world. Alice in Wonderland delusions are where people wake up feeling huge. They're not huge, but they feel their body to be massive. It might be their whole body, that the room is small, but they're massive. Or it might be that their arm or their hand is now huge. So they suffer. These patients normally have perhaps a tumor or some, in the case of Alice in Wonderland delusions, it might be some psychiatric disease. But the idea here is that you can see these distortions of space. And so the idea in neurotopographics was to present not just what we see that we do with the rats, is to record somebody's position in a room being chased and mapped, and hear the cells that you heard bursting away, and map the potential structure of the space. But let's now, because this is a piece of art, we can go beyond just simply illustrating that by showing the subjective experience of the person walking the room. And in particular, in this case, this is someone who is struggling with reality. They're suffering hallucinations in the space. And so shown here is the path that man takes through the gallery, and the mapping of the space, the grid patterns into the space, the objective view of the man as he walks through the room. But what's shown here, and I'll zoom in now, is some views cut from the movie that I couldn't show, unfortunately, of where his view changes. Things start appearing in the walls around him. The world stretches and alters, and things appear. And the idea there was to get a mapping between the activity cell patterns and the internal state of the character walking around. It's a way of exploring this and seeing what happens. Of course, the audience who've stumbled into Gimple Feast don't necessarily know all of this backstory. If they took the trouble, they might start reading about the information sheet. They might go onto web. They might start discovering, whoa, this is really strange, these grid cells, and discover, in fact, maybe in the narrative of that film, there was not just a grid cell, but there was a place cell hidden in there. I don't know if anybody noticed the hidden place cell in that movie. I doubt it. Anybody would have spotted that. But this was a particularly important manipulation, I think, for exploring that idea. So with that film as well, we thought we need to come up with a narrative as well to get that movie into place. And the man enters the room. He has to map the space. And at the time we were making the movie, the science was moving. And literally, as we were devising our film, researchers were publishing in Nature Neuroscience ideas of stretching in the grid. That if you enter a room, if a rat's in a room, that if it thought initially it was a square, it turns out to be a rectangle, the grids stretch. And there'll be more data next week at the Society of Neuroscience on stretching grids. We're still trying to understand it. But we thought we'd play with that idea in our film installation. So sort of provoking ideas about the science. There's another idea in the place and grid cell literature, the idea of maps, remapping the space, and as the man walks through the room, his map might stretch and it might unanchor from the space around him as he suffers these delusions. And these ideas from the science all drove into how we created that final piece of artwork, though it is not explicit in the actual narrative of the movie when you visit it. So that's neurotopographics. That's how that project developed. And there were some good things and some bad things. And I spent 2 years learning about how to explain grid cells and place cells to Anthony and Bettina, who had no scientific or not much scientific background. I learned a lot in that time. But I thought at the end of all that, we'd capitalize on that and develop this next project. So pattern completion is another project. We didn't go for the sort of sexy words using the Latin or anything this time. We just went for the simple name. Pattern completion of this project refers to a particularly theoretical mechanism scientists have long believed exists in our memory system. And I'll come on to that later. But this is a project by Michaela Nettle and Tom Simmons. Michaela and Tom, Michaela is a visual artist working in London, and she's married to Tom, who's a sound designer and is also a senior lecturer at the Royal College of Art. And the idea from this project sprang out of visiting one of Michaela's exhibits, where she had developed this method of projecting into glass spheres. I mean, there's a lot of work on projecting into glass, but this was a particular exhibition she had developed. There's a single glass sphere of somebody swimming through a pool, and I thought, well, let's see what we could do with that. Is there something we might do to use that projection to play with the ideas of memory, how our memories operate? And let's involve Tom, because maybe we can use the sounds. So pattern completion, rather than having one sphere, we developed a set. We used a set of spheres. So here is a large-scale slice cut through the brain of a primate brain, and this is the hippocampus here. So that's where John O'Keefe discovered these place cells. Here's the entorhinal cortex, which contains the grid cells. And this project pattern completion really works on what are these cells here doing, and taking the ideas out of the neuroscience. And what I really wanted to do with this project was take the ideas out of the lab, out of those papers, and play with them and explore them. Generate something engaging and evocative and interesting that really touches on the edge of where the science is. Not what we know, but what we think we might know. So these cells in this region of the brain, if you damage these, if something goes wrong, you don't get enough oxygen, or you get encephalitis and they die, you'll never remember anything again that happens to you. We know this also from surgical work. This patient, HM, suffered bilateral lesions due to a surgeon removing his hippocampus, and HM never remembered from the age of 25 to 80 any new experiences that happened to him. So something about this network is doing that function of recording, storing memories. And the idea in pattern completion was to look at the theoretical idea underlying that. Neuroscientists had wondered, how is it that these cells store the memories? What do they do? And you can see here in the stain, these little black dots are the bodies of all the cells that sit within this layer of the hippocampus. And this particular part I'm pointing to here is called the CA3 region, the cornus albuminus 3. And it's a particularly important part of the brain in the hippocampus because the cells here are highly reconnected to each other. And that'll become important as I go into the next bit. It's a particularly unusually interconnected bit of the brain. If we zoom in on one of the, if we zoomed our camera in on one of these little dots and filled it in and looked at the whole structure of one of these little dots here, you'd see it look like this. This is a pyramidal cell in the hippocampus. And the pyramidal cell in your hippocampus will look very similar to the pyramidal cell in a bat or a rat or a cow or any other mammal. They're very, very similar. Nature has not tinkered around much with the hippocampus. It's the same structure, whatever animal you look at. It's a very important old structure in the brain. But if we want to think about these cells, of course, we can look at the physiology, we can look at how they operate, but we can try and also build models, computer models, of how would these cells operate and simplify it down and think, what are the fundamental mechanisms? What is the properties of these cells that allow us to remember the past? One way to do that is simplify these really elaborate systems down to this thing. This is a computational unit in an artificial neural network. So here it's got a little pyramidal. Here's the body that decides whether the cell is going to become active or not. Here's what's known as the dendrite. These are the tree-like structures that come out of the cell, the dendrite. And here's the bit of the cell that sends a message out. So this is like the gathering part of the cell, the information gathering bit, and the output, the axon. And of course, there's more than one. You have to have a network of these cells, all projecting information out. And as you heard earlier, if these are in the hippocampus, they're likely to be place cells. This information is a continuous output of where am I? Where am I in space? Or where would I like to be in my mind if I imagine my bedroom now? Those cells are the ones in my bedroom that are active. But what's going into these cells are inputs. So we might have some inputs coming in from one area of the brain and latching onto these cells. These are synapses connecting up cells that are projecting in here. And we might also have inputs from another part of the brain. So multiple brain areas converge. And in fact, the hippocampus is not only interesting because if you damage it, you lose all your memories, but also it's one of the latest part in the brain in terms of processing stages. You get all the visual information from the world, all the auditory information, all the tactile touch. All of it goes through lots and lots of processing before it arrives in the hippocampus. It's really at the high level of your brain in terms of constructing something. It's not at the sensory surface. So these are highly developed processing ideas. This might be, the idea, it might be a particular face or a person that's coming in. But we can also very simply, rather than getting into complexes of the physiology here, we can imagine that the inputs into these cells, this particular network, are binary. The cells really pulse either on or off. And so the inputs might come in where this input here is active, this one's active, but these two aren't. So these are silent, but the activity pattern coming into these cells is a 1, 0, 0, 1. It's just an arbitrary pattern coming in. And on this side, you might have another pattern, a 0, 1, 0, 1. So we can just simplify these cells down to converging patterns. Now, according to the laws in these networks, if you took these two patterns together, and these cells need to summate the information coming in, you can see that this cell would become active because it's getting excited by this cell and by another cell sending in here. Two cells are driving it, and it's enough to evoke that cell to start waking up and firing. These other cells aren't getting active because they're not getting enough information to drive them. So with this set of inputs, this might be imagining my bedroom, and the cell that likes my bedroom is now active in this network. But the key, and so it's the output comes out 0, 0, 1. So these are just patterns of activity. But the key idea in neuroscience goes into memories in terms of networks and what people think is going on, what forms a memory, is this. The idea that these connections between cells projecting into the system is that they're modifiable. Something known as synaptic plasticity. These connections, these synapses between the cell can change. And what's happening here in this network is that these cell projections in here are changed so that it becomes easier for this cell, this projection, to drive that particular cell. And so what that now means, if we look at this particular input pattern where only this activation is coming in here and nothing is coming in the bottom, previously none of those cells would have been active. They wouldn't have received enough input. But now, because of this change, there's been a stored change in the synapses. Now that's sufficient for this cell to become active. So the idea here is that restoring the conjunction of the smell of a rose and the picture of a rose, all you need to do to remember the smell of a rose is see a picture of it. And it can re-evoke rose in your artificial network. This is the simplistic way in which scientists think about these patterns of memory. So the idea of pattern completion goes a little beyond that, because the idea of pattern completion comes from the idea that this network talks back to itself. The network doesn't just have stuff coming in and shooting it back out. The network sends out information back through itself, and it modifies the connections within the network. And that has a very interesting property. That means that as these cells become active, they change the activity patterns of other cells of the network. They reverberate, they reiterate, the pattern changes, and it can change and fall into certain states. So if you look in the literature on pattern completion, this idea of our memories being patterns that need to be completed, the smell of a rose needs to be completed by seeing a picture of a rose in my brain to really re-experience a rose. And the idea is that these networks can store patterns like this as connections. And so what